So it's been called the final frontier, space, that is, and we're hoping that Dr. Joseph Borg takes us where no speaker has gone before, in this case, space omics. Joseph is an academic molecular geneticist and the principal investigator at the University of Malta. Primary interest is the understanding of developmental globin gene switching, regulation and control of erythropoiesis. I'm not even going to try to do that one again. I'll let you correct me on that. And the next generation sequencing tools to uncover locked secrets of life. He recently led Malta's first space program for life science in space through the Maleth Program. And he also actively participates in the analysis working group at the NASA GeneLab for multi-omics, and on the Topical Team for European Space Agency Space Omics. Joseph, my apologies for the Star Trek introduction. I'm sure I'm the first person that's ever connected your name with that, right?
Yeah, indeed. Do you hear me correctly? I hope you do.
I can hear you just fine. And if you want to go ahead and go to slideshow mode, I'm going to get out of your way. It's all yours.
So thanks so much to the organizers for the invitation, which I gladly accepted, and I feel really, how to say, honored to be part of this Omics research symposium, because indeed omics is one aspect of my research work that I really enjoy doing. So yes, so background aside, and focusing on blood research and blood omics. Today I'm talking to you about Maleth Program, which is a life science research program in space. And this is centered around diabetes type 2, with special interest on human skin microbiomes that are found on diabetic foot ulcers. And I will explain why we study these in the context of space, on Earth, and how we compare the two with each other. So as a brief intro, let me just minimize the view screen. Yeah, so all of this, the research program, the Maleth program, started at the end of 2020. So during the pandemic years here in Malta, I had an idea to sort of conduct or take my next level of omic science in the context of space. It all came about serendipitously over the internet, over LinkedIn, having the idea and then meeting a group of people that can actually bring the idea to fruition. And at that time, I had taken the idea to our Ministry of Foreign and International Affairs. The idea being that sending an experiment to space could not be more foreign or more international than this. The idea was well received. And from there, basically, we continued forth with our program. So a very quick rough shot about the program. So we did a number of pilot studies in our labs on Earth in Malta to ensure that the human skin tissue samples, the way they are preserved, the way they are prepared in buffer, that the bacteria, the whole microbiome, is maintained for a number of weeks or months, and they do not lose their viability, and that we are able to both culture them back on agar plates, and also we are able to extract DNA and test it by NGS, mostly 16S typing, to determine the classification and the taxonomy of the bacteria. This project ties in very strongly with an ongoing PhD program of my student, who is testing roughly 150 or 200 diabetic patients from Malta. So we only had a selective number of patients that we also progressed to the space study. This work was done in Malta and also in Belgium, where we assemble our so-called biocube. I will show this later on. Basically, the cube itself is shipped from Europe to the States. From Cape Canaveral, there it is assembled on a SpaceX rocket. SpaceX, as some of you or most of you know, travels to outer space, and in this case, docks with the space station. Once it is present or docked with the space station, the astronauts get to work. They take out the cube, they assemble it to a particular place where the cube is powered up. And from here, from Malta, from our control room or center, we are able to connect to the cube and follow the experiment, so to say. Broadly speaking, the experiment per se is not too exciting to monitor, because I mean, the bacteria and the skin tissue don't really change macroscopically or visually. except that you see everything floating around. But the real work is done when the cube is returned back. That is what we did for Maleth 1. And for Maleth 2, which is a follow-up and a replica, because as all of you know, in science, you cannot really depend on one experiment and biological replicates, but you also need to validate your findings. And that is Maleth 2. And Maleth 2 is right now on its way from Orlando to Europe and on its way back to Malta this very week or the end of this week. So it's an exciting time as well. Okay, yeah. So of course, all of this precisely fits in the context of science. I form part of NASA GeneLab as one of the working groups. Also, ESA, the European Space Agency, also has its own space omics team. And basically, in a nutshell, what these groups are is the experiments that we send to space and back, whether they are animal models, whether they are bacteria, whether they are plant or humans, astronauts. The biochemical and genetic data that we get back are deposited in these databases, especially the NASA GeneLab repository. And there the data forms part of open science. And we call this open science for life in space. We published a white paper a while ago where it sort of drives the message home about how we can take space biology to the next era by ensuring standardization, applicability, and also a number of important tissue analysis and fixation protocols. We did already have a number of scientific publications on this topic, not necessarily on Maleth, but it also feeds indirectly. And these fall under the cell press umbrella. So it is an area, it is a topic where it is very well sought also for publication purposes. So this is Maleth 1. This is the first cube of last year. It is a 3D printed cube using special plastic. Those are the logos. And what is Maleth 1 basically? So Maleth 1, as I said, it centers around diabetes. Diabetes, as you know, is a major public health concern worldwide. And Malta, the country that I live in, is one of the highest prevalent in Europe. Actually, about 12% of the whole population is affected. A large number of diabetic patients are at risk of developing foot ulcers, which is a major cause of concern for hospitalization. There are different types of signs and symptoms. Generally, the people, a major problem is that they do not speak up, and therefore the infection and the wound ulcers tend to become more serious by time. The infections are polymicrobial, so you cannot pin down one particular microorganism. And a large number, almost half or more of these infections result in osteomyelitis and also amputation. In Malta alone, in 1 year, there are 400 cases of minor to major amputations. So more than one person per day, which is a very, very significant number. So this was the idea behind a PhD study, which is ongoing, and also the idea to marry this together with a space science research. This is a very conservative diabetic foot ulcer. Generally, they are much worse, much bigger. And yes, here I would like to mention my PhD student, Christine Gatt, who's a biomedical scientist. She works in a laboratory at our hospital, and she's conducting her PhD work at the university. And she's studying the genetics of bacteria that infect wound ulcers. Like Christine, I have a team of at least 3 other students one is also doing a PhD focusing on ophthalmic disorders, and 2 master's projects which are focusing on globin gene switching and developmental control of hemoglobin. So the way that we work as a team together, even though the students are not all on the same topic, we share the experience, we discuss with each other, even in terms of journal clubs, and generally this yields a rewarding and a rich experience. So this is a typical look inside the BioCube. You can see half of the cube on the right-hand side is focused with a Raspberry Pi and computer essentials or accessories. And on the left-hand side is the unit where there are cuvettes ready to be assembled with the skin tissue sample itself. Something small aside, I am a strong believer of training and inspiring the young generation. And so since in the BioCube we had space. We had an SD card that can be utilized to embed messages or artwork from school children. We did this. We invited school children from Malta to have or to draw something that has to do with climate change, global warming, or send a message to the world. And we embedded all of these files inside the cube in such a way that we can open them when they are in space and stream them or show them on social media back on Earth. And this was a nice exercise, and we could see that the young children were really enticed. So this is just two quick pictures about the sample preparation. And on the right-hand side, you can see the samples being integrated. And this is a more closer look at the human skin tissue samples and how they are placed in each cuvette. Okay. Of course, the BioCube is one part of the experiment. On the other side of the story, that is on Earth and in Malta, we also require a laptop or a mission control room where we can plug in our system, and we are able to communicate or contact directly the BioCube itself for many reasons, some of which are for aesthetic or fun, the use of camera to see the samples to ensure that there were no major breakages or problems. There are also a number of sensors to detect the temperature, humidity, and one can adjust those sensors. And there is also LED lighting, and that you can change different colors according to the mood that one is feeling. But basically having a mission control center back on Earth ensures peace of mind that the experiment is going well. This is the cube itself positioned on a module, which is an exact replica that is found on board the Columbus module on the space station. Okay, IceCube stands for International Commercial Experiment, and you will see one of these also on board the space station later on. This is the cube in its final steps, with all the samples integrated, packaged in a special box, and shipped to the States. And from the States, as I said, the astronaut handled the cube, and you can see here.
The station to the IceCube activity is complete. Now looking for the pre-conference. We are very much grateful for your support today. We also took note of your juggling skills, so thanks for the entertainment. And the pre-conference is ready on Space to Ground 4.
So there's just some talk between the astronaut and mission control.
It's good to see those new players in space, Malta and others. So congratulations to them for their success and moving forward.
That's when they mentioned Malta for the first time. And there you can see our cube alongside others placed on board the IceCube's facility, which is located on board the space station. Once plugged, that is powered up, and we have access to it from Earth. Once roughly between 30-35 days elapse, the same spacecraft that took the experiment to the space station is undocked. It deorbits around Earth and falls or splashes down in the Atlantic Ocean. There, a boat will go out, it will retrieve the spacecraft. The spacecraft is taken back to the Kennedy Space Center, and from there, all the experiments that were sent from the space station back to Earth are sorted. It's just like a postal service, and we receive the cube back to Malta. And there where the real work starts, where we open up the cube, we take out the samples, and there is a battery of tests that we do on all these skin tissue samples, which includes aerobic and anaerobic bacteria, the use of culture media as broth, and also culture media as agars, and various forms. Of course, Christine Gadd, being the microbiologist, is much more well-versed in all the different tests that we do. An interesting observation or a photograph that we had to do was when we identified that bacteria sent to space and back were actually cultivated on agar plates. So this showed us for the first time, for us in Malta, that we were able to send an experiment. The bacteria and the skin microbiome survived and came back to Earth to sort of tell the tale. Yes, so there's a large number of genetics and omic tools that we are doing The first of which is concluded, is a first wave analysis, which we call a 16S typing. And 16S typing serves as a taxonomy classification of all the bacteria that are present in the samples. In the Maleth one, we had 6 different samples. So 6 different patients that came to our hospital, and each patient, a human skin tissue sample was obtained. The human skin tissue sample, as part of the surgery process, was split in 3. Okay, one was immediately processed and frozen. So we obtained a human skin microbiome profile on day 1. And the other 2 pieces, one was left in our lab on Earth, and one was sent to space and back. And therefore, for each single patient, which we have 6, okay, we have 3 different data points, as you like. Okay, so we have the Omics or the 16S typing for each of the 3 timelines, and we can compare the differences between those 3, and we can also compare the differences between the 6 samples with each other. Okay, and there where you obtain a comparative genomics approach. This figure shows you a spread and the largest number of bacteria found together with the lowest number of bacteria found in the skin sample itself. Of course, this was replicated for all samples. Yeah, so our aim was to obtain a signature that is correlated with the tissue type itself. We had many other tissue types as well. I mean, we had a simple skin swab from healthy controls to act as a control. We had the foot ulcers themselves, those which were sent to space station and those which were left on Earth. We also had biofilms from certain wound ulcers, and we also had negative uninoculated controls. There is extensive informatics that was done. I did not do this myself, but a team of bioinformaticians, also with the help at NASA GeneLab, which exclusively work on these type of analyses. And interestingly, from these analyses, before going into metagenomic sequencing, which is being conducted as we speak, so to say, which we are waiting for the Maleth 2 samples as well. But from just the 16S typing, we could see, or we could observe, that a small select number of bacterial species in space appear to thrive or flourish more than the same tissue type that was left on Earth. So there are these subtle differences, but meaningful, between one tissue type of one patient left on Earth, sent to space, and the differences between the 2. And we have a select number of bacteria that exhibit these differences. We can mention that later on. One key bacteria is Morganella morganae. Yeah, so the immediate next steps is that we are comparing the 16S data between these 3 data points over the 6 biological replicates. Something which we haven't done yet, but we want to explore, is that the patients themselves who came to hospital, we will also be sequencing the full genome of each patient, and this we believe will give added data, added information that we can sort of interplay or overlay, so to say, the genomic data of the patient together with the microbiome data obtained from the human skin. Metagenomic sequencing is a no-brainer, so that is being done on the isolated microbiome together with colleagues in the States. And of course, there are a number of key people that sort of what I have shown or presented in these few slides have made this possible. So primarily, there's the Ministry for Foreign European Affairs, and for Maleth 2, there is also the Ministry now for Research and Innovation. A number of key people that are in the States, and these are Chris Mason, is a very renowned scientist who in the past has worked on NASA twin astronauts. Maybe some of you know Mark and Scott Kelly, one of them is a senator. They are twins, they are identical twins. They are both astronauts. And there was a huge study where one of the twins was on Earth, the other twin was a year in space. And therefore, there are many blood samples being taken, urine samples, and other measurements. And one can conduct all the genomics and the multi-omics that one can think of, and compare between the two. And it's amazing the results that one gets back. Braden Tierney is the one who conducted the informatics for the Maleth program. Afshin was the original point of contact at NASA, who introduced me to the world of space bioscience. I am indebted to him. Kevin Kassar and team, he is a vascular surgeon at our hospital, and he's the one who physically operates and takes the skin tissue. I mean, we depend on these medical professionals. Arcafort and Evolve are private tech companies. It's impressive the interest that we see in such experiments or programs by the private sector who want to be part of this mission. Also, an interesting bartering idea, as if you like, is that the biotech companies that agree to sponsor our work or sponsor our mission, they get to emblazon their logo on the cube itself. So that is exposure that is out of this world in the literal sense of the word. And of course, Christine got this. She's the PhD candidate who's doing all of the groundwork in the lab, physically working. Yeah, so this slide is, we're almost at the end here. The slide is sort of a collection of Maleth 1, which is the top left cube, and Maleth 2, which is the bottom left cube. You can see a color change. Maleth 2 is an improvement on the cube design. It is made up of a machined aluminium. So this is like the same as a MacBook laptop or these new fancy laptops that are made of aluminium. And basically you have a block of aluminium and they are machined from the inside to outside. And there was also an upgrade of the Raspberry Pi computer itself. And there on the bottom, on the center bottom, you can see a USB dongle, a key that is used to connect directly with the ISS. An interesting idea or notion is that once you plug in this dongle, okay, automatically the mission control room or the laptop that you're using disconnects with the internet and you only connect with the ISS. This is part of the encryption and security policy. And on the far right there you can see the SpaceX rocket that departed Earth to arrive to the ISS. This is a list of all sponsors and logos to date, and 3 pictures that shows you a close image of Maleth 2 cube. Yes, Maleth 2, I didn't speak too much about this. We had 5 samples and not 6 microbiomes, and we had 1 sample reserved for dried yeast, Saccharomyces cerevisiae. We have some ideas that we wish to implement when the experiment gets back. And as the cube itself on board the platform itself. And I believe that was the last slide. And if you have any questions or ideas, or maybe you have an idea to send to space, feel free to reach out to me on joseph@spaceomics.com. And watch out for some emerging publications on Maleth. So with that, I thank you all for being patient to listen to me. I think I cannot hear you, Grant. I cannot hear you.
Good old microphone muting, right? Does gravity play a role? This is from the audience. Does gravity play a role on aging process and the rate of cell division? I'm assuming he means in space, so in gravity. Yeah. So does gravity play a role?
So there are some conflicting, of course, this is a little bit different from the Maleth program that we do, right? However, in the context of aging, we have been told that in space we age faster. However, recent evidence, and it's also published, there seems to be observation that telomeres at the end of the chromosomes, instead of shortening, actually in long duration they elongate, and that means that the cells might actually become more younger. So maybe we'll be having trips to space to become younger as opposed to aging.
Well, that'd be interesting. A little expensive, but interesting nonetheless, right? So how is the Maleth program used to understand living in space?
Yeah, so Maleth program, okay, so it's ambitious. It's a very first program for Malta. And the idea or the overall objective, besides the core is a life science experiment on diabetes type 2. We also know that astronauts in space generally are very healthy and athletic. Okay. However, astronauts in space also have difficulties with vascularization and the bloodstream itself. They endure space anemia. So understanding wound ulcers, foot ulcers, and the complications that arise on Earth can feed in directly with complications that we observe in space, especially if space is opened up wider for astronauts who might not be completely athletic or fit.
Like some of the people that can just pay to get there, right? So do you think there are specific measures that astronauts take before or after going into space? Maybe talk about one or two of them.
This is a very, very good question. I think to date, it is still not completely set in stone, but there are definite measurements. I mean measurements that are done at the brain, the EEG, and cardiac function. I mean, these are logical tests that need to be done to ascertain that the individual, the astronaut, is fit, especially to survive a 3-month, a 6-month mission. An interesting thing is the introduction of omics testing, genomics, DNA, RNA expression profiling. This is a very, very, very recent entry. And as we progress, we're going to see more and more omics testing on astronauts.
Fascinating. Well, thank you yet again. This has been wonderful. I apologize again on all the Star Trek references, but that was fun. Glad you played along.
Thanks.
You're welcome.